Hello everyone, my name is Frankie and today I'm going to present the journey of drug discovery. The drug development process is a massive undertaking. On average, it takes 12 to 15 years to bring a single molecule to the market. The financial investment is equally high, often ranging of 1 billion to 2 billion US dollar. And most importantly, we must respect the attraction rate out of 5,000 to 10,000 compounds that enter the pipeline, and there are only typical only one attempts final approval. The process of drug discovery begins with identifying the biological origin of a disease, such as a specific gene or protein. Then we must then validate this target to ensure it is druggable and that modulating it will actually lead to a therapeutic effect. This stage is critical because it prevents us from wasting years of research on a biological pathway that doesn't actually control the disease. Once we have a target, we look for a lead, a synthetically stable molecule that shows high specificity for that target. We conduct a druggability assessment here to evaluate the molecule's potential to bind and its metabolic profile. This stage effectively narrows down the millions of potential molecules into specific chemical class that shows preliminary evidence of working. In step 3, we use iterative synthesis to refine the lead compound. As chemists, we will alter the molecular structure of the drug to improve its potency and selectivity while reducing the deficiencies like high toxicity. The goal here is to produce high-quality candidate that is ready for animal testing. Before human testing, we must evaluate safety and efficiency in animal species. In this stage, we will look closely at pharmacology, focusing on ADME, which is absorption, distribution, metabolism, and excretion, and toxicology to find unwanted effects. This successful data here will allow us to file an investigational new drug application with the FDA. And now we'll go into the clinical research. The clinical trial occurs in four main phases. The phase one is to test the drug safety and dose in 20 to 80 healthy volunteers. And the phase two is to expand to a few hundred patients to evaluate efficiency and side effects. And the phase three involves thousands of volunteers to confirm the action benefit and detect rare side effects. And finally, the phase four is post-marketing surveillance to monitor long-term safety in the real world. Nowadays, we are using several modern advancements that are transforming the drug discovery process. Firstly is the computer-aided drug design, which is using software like QuickDrop to predict drug behavior before we ever synthesize it in the lab. And the structure-based design and matrix is using X-ray crystallography to help us to identify the exact small molecule that fit into target receptor. And the new metrics like ligand efficiency is to help us to ensure we are designing rational molecules rather than just sticky ones. And lastly is the biologics and personalized medicine. This is a major shift toward monoclonal antibodies and the use of biomarkers. This allows us to practice personalized medicine matching the right drug to the right patient subtype, which will significantly reduce the failure rate. And here we come to the end of my presentation. Thank you for your attention.